So nice to meet everybody. My name is, as was already mentioned, in Dutch Leo, but you can also call me Leo Marcelis. I'm a professor of horticulture at Wageningen University. So I work on horticultural crops, meaning in particular crops grown in greenhouses or vertical farming. Yeah, and then today a bit focusing on photosynthesis. I will not dive deep into the photosynthesis, but more a bit on the application of it in the greenhouses and vertical farms. So, looking at greenhouse production over the years, we have seen a huge yield increase. So what we see here in this figure is the yield, and then for cucumber, tomato, sweet pepper, usually we express it in kilograms per square meter per year. Well, for the green is then the cucumber, you see two lines, and the same is for tomato, red, two lines. Well, there is a broken line. The broken line is that for growers who use supplementary light. So that nowadays most cases LED lighting in more recent years. Also for tomato, and what you see, tomato is getting about with lighting 90 kilograms per square meter per year is already quite a normal production. So huge yield. How do we get it, and how important is photosynthesis? But also this improvement over the years. Why is that? Is that due to breeding? So you could say roughly in 30 years' time, the yield doubled. Any idea on what part would be due to breeding? Well, it's difficult to make an estimate, but what a colleague of me they do, and I see here one experiment, they do in fact a few experiments growing cultivars which were released in different years, all under the same conditions. And that's of course then a drawback, but if you grow them under the same conditions, you can roughly see what is a newer cultivar more productive. Well, roughly you could estimate, and just a very rough estimate, about 1% yield increase per year is due to the breeding, which means there is still 70% of other factors. But then looking at those cultivars, why are more modern cultivars more efficient? If you would ask it to people in corn or wheat, where there's also a lot of yield improvement, probably they will tell you it's the assimilate partitioning, the harvest index. And my initial work was all focused on partitioning, and I would assume it is like that. But no, it's not. And I do not show you here all the results. But what became quite obvious that the cultivars, the newer cultivars that give more yield, that also correlated quite a bit with leaf photosynthesis. And that is what we see here in the left graph. We see here the rate of leaf photosynthesis of the different cultivars, and you see a reasonable correlation with the more modern cultivars had a higher rate of leaf photosynthesis. So yes, there is a correlation between higher leaf photosynthesis in newer cultivars and their yield. Another factor that seems to play a role is the light extinction coefficient. For those less familiar with it, it means if the coefficient declined, which means the light penetrates deeper into the canopy. Well, if you have a canopy with sufficient leaves, it means the same amount of light is more or less the same amount of light is intercepted, but distributed in a better way. So also that is an important aspect. Okay, so related to photosynthesis, yes, it seems that photosynthesis is relevant for yield for the modern cultivars. And in general, I often get the question, oh, let's improve photosynthesis and then we get more yield. Or I get the question, do we really get more yield? It all depends on the balance between supply and demand of assimilates. So supply of assimilates, that's roughly the photosynthesis. That's what we see here on the left-hand side. Photosynthesis, of course, it depends on the leaf area and climatic factors like light in the first place, CO2, temperature, air humidity. Then we get production of assimilates. That's where the plant can grow with. Those assimilates can be used for respiration. They can be used for growth, for structural dry mass increase. Whether those assimilates are being used for growth, so in converting the assimilates in structural dry mass, well, then we need to have zinc organs on the plant. Temperature plays an important role on that, and that needs to be balanced. And only when it's balanced, a higher increase in assimilates will work in increasing the yield. I often get the question, in greenhouses, we may use lighting. We get more source, so source here then another term for the photosynthesis. Does it lead to more yield? Well, If there are, and then I take here tomato as the example, but it holds for all type of plants. If there are sufficient sinks, so sink organs that can use the assimilate, it will lead to an increase in crop production. However, if there are not so many sinks on the plant, yeah, you can produce more photosynthesis, you can have more photosynthesis, but the photosynthetics can't be used. And in those cases, you can give more light, but it will not lead to more yield. Okay, so let's now take a look at tomato plants. There's a lot of things behind this graph, and I don't have the time to explain how we did all those estimates exactly. But what we estimated, the source-sink ratio of the tomato plants. So supply of assimilate versus the sink demand of the plant. We did do that for, you see, 3 lines. That is for 3 different tomato cultivars, one which has typically a large size of fruit, a medium size, and a small size. And it was assumed, well, small tomato plants with small fruits, that are small sinks, so maybe they are they are more sink limited. Well, it is the first days after planting, it's in the green, so the results will also very much depend on the season. But what we can learn from this graph is that here in this gray area, that is when the source-sink ratio is larger than 1, the plants are sink limited. Typically with tomato, that can occur in particular with young plants when there are not yet the fruits on the plant. However, if we then go once the fruits are on the plant and if we grow them like a commercial grower would do, and then based on this experiment and other experiments, we always see that tomato is source limited. The sink, they have so many fruits who could use the assimilates. Normally, they are, if we grow them in a commercial way, they are source limited, which suggests that if we can increase the photosynthesis, we can increase the yield. But the opposite is true for the young plants. And if we look in academic research, we often work with young plants because that's much easier to work with. And often we grow single plants, meaning not as a canopy, but at quite a bit of a distance, which means if there's not directly a neighboring plant, the plant gets in fact quite a lot of light. So I see often research on young individual tomato plants, which are in fact sink limited. And then care is taken if you want to extrapolate those results on those young plants to a fruiting plants which are grown as a canopy, with a high density. Okay, always important, keep in mind what the source-sink balance is if an increase in photosynthesis can work. Now going a bit towards the greenhouses, but the same would hold for vertical farming. Energy use is high, but there's no other conclusion that we will go towards climate neutral greenhouses. In the Netherlands, the target is that you are climate neutral by 2040. And a large part of the energy is used for the lighting. So that's why a lot of our research is focused on how can we make the lighting more efficient. And there are 3 steps. First is reduce the energy use drastically. But of course, we still want to have a high yield and a good quality. And when we reduce the energy, the next step is we will never manage that no energy is needed. There will always be energy needed, but then let's use only renewable energy. But if we look at the price of energy, it's fluctuating from day to day and within the day. So let's use it when it is less expensive. But how to do that then? Okay, and then we can look at the lighting, making smart use of the light. And if you think about light, well, then we first need to consider light has many different aspects. It is the intensity of light, which also gives the light intensity directly relevant for photosynthesis. There is the spectrum, so the color of the light, which can have an important impact. It may be about the photo period, and some plants, maybe we can grow them under 24-hour lights, and some other plants can not at all grow under continuous light. We should also consider the direction of light, where the solar light always comes from the top. But if you're thinking about LED lighting and a tall crop, well, why should it only be on the top? Maybe we can have that in between, and then we call it interlighting. And light also represents energy. well, it has consequences for transpiration, has consequences for the whole energy balance or heat balance of the crop. Well, then the same picture, but now a little bit extended. It is light has the different components, but it also affects many processes all at the same time. It has effects on photosynthesis. It has effects on the plant-water relations. It has effects on the morphology on the plant, on the partitioning of assimilates. And also those processes affect each other. So I would say, as a plant scientist, that's really nice because then we can study that all in their interactions. And by the way, also, all the light effects usually show strong interactions with other growth conditions like temperature, or the CO2 concentration, or the air humidity, or the plant nutrition. Let's first look a little bit at what are the effects on photosynthesis. And what we see here is the relative quantum yield of photosynthesis plotted versus the wavelength. Let's first look at the blue line. That's probably a curve that most people recognize. Usually, if it is measured, it's measured on a single leaf. Well, then usually what we see at red light, there's a peak in the photosynthesis. There's a bit of a dip around the green and a smaller peak in the blue light. So that's the blue line if you measure on a single leaf. However, please realize that we will never grow one single leaf. We also, as a grower, will never grow a single plant. It will grow a plant at a high density, where there are many different leaves at the same time. And if this whole canopy of leaves consists of the same leaf, which with the characteristics of the blue line, we get the red line. That's the relative quantum yield of the crop. Relative to what it is for the red light. Well, we can say it is more or less the same curve, but we can also see that where there is for the single leaf quite a bit of a dip in the green light, for the crop there is less. Well, why is that? Well, it's easy to explain. On a single leaf, quite a bit of the green light, or a larger fraction of green light, is not absorbed, meaning it is reflected and transmitted. If it's not absorbed, it's lost for the leaf. But on a canopy level, transmitted light is not lost because maybe this leaf transmits some of the green light. Well, then it will be absorbed by the leaf below it. So that's why there is a difference between a crop and a canopy of leaves versus a single leaf. I would say measurements on single leaves are very valuable, and we know what it means for the crop as a whole. We have simple calculations for that upscaling. So what we see here, red light is the best for photosynthesis. So then the question comes, should we grow then the plants only under red light? Why not? Photosynthesis here highest. However, growing plants under only red light usually is not a good solution. That's what we see as an example in this curve, where you see on the y-axis the A-net, the net photosynthesis of cucumber leaves. And on the x-axis, we see the irradiance, the light intensity for plants grown for a longer while under different fractions of red-blue light. It's not necessary that you look at all the different lights. You can see the lower curve, that is for plants grown under sole red light. So the previous slide, which was direct effects of light color, red is the best, but if you grow the plants for a prolonged period under only red, they do not perform well. Usually what we see if we grow plants under one single color, we get a disturbed functioning of the plant. Not always, but many times we get a very different type of response of the plants. Also, if you see research results on one single color, be careful on how to interpret it towards mixtures of light. Then let's move on to another color, the far red light. Far red light, usually defined as 700 to 800 nanometer or 700 to 750 nanometer. It's called far red. Some people say it is dark red. Yeah, you could call it like that. Strictly speaking, we hardly see it with our human eye. It's just a little bit beyond. There is the PAR light. PAR stands for photosynthetic active radiation. Classically, PAR is defined from 4 to 700 nanometer. Far red is not part of photosynthetic active radiation on how it is defined. However, and that is based on initial work where if you give only far red to a leaf, there's no photosynthesis. However, If it is in combination with other colors, far red is also photosynthetic active, and in particular, the research group around Bruce Bugbee, Xuan Zhen, and more people showed far red is photosynthetic active if it's in combination with other colors. And they claimed it's in fact as effective as classic PAR. And they said, well, we should not only have PAR, but we should use the term EPAR, extended PAR, which includes far red. Okay, very interesting. So we also studied that a bit more. And yes, I would conclude far red is indeed also photosynthetic active. And that's what we see here in this graph. This is maybe a little bit of a complicated graph, but I will try to guide you through it. Do you see also my mouse? I'm not sure. So we'll see on the y-axis the net photosynthesis rate. On the x-axis, the incident TPFD, that stands for total photon flux density. So all the photons from 400 to 800 nanometer. If we start on the left side of the curve, where we see the first points at around 200 micromole, and first look at the black curve. Black curve's going up. That means we start at 200 micromoles, and then we add 25 or 50 or 100 micromole of PAR light, in this case red, blue light. We see photosynthesis increasing. Then we do look at the red curve, starting at 200 micromole of PAR light, and then adding not PAR light, but the additional light is then the far red. So adding 25, 50 or 100 micromole, we see the curve going up. So what we can learn from it, the red line is going up if you go to higher photon flux densities, meaning there is photosynthetic active. But we also see quite a clear difference between the red and the black line. So I would say if you look at efficiency, well, the efficiency of far red is not as high. This all is based on measuring the light intensity just above the leaf. But now let's look on the absorption. So this is the same data, but now on the x-axis is about how much of the light is absorbed. Then, wow, this is interesting. Now we see the red symbol, which are with far red, and the open symbols, the black symbols, the white symbols, that's without far red. They're all more or less on the same line, indicating that far red is as effective as PAR on the basis of absorbed photons. However, far red is less absorbed, and because it is less absorbed, it's on an incident basis less efficient. So that's the main message that I would like to conclude about far red. But please note, far red, this is on photosynthesis. I would say the even more important or bigger effects of far red are on many other growth responses, the morphology, all processes mediated by phytochromes. Then we can also think about what I said, well, the direction of light. Here we see a greenhouse for tomato. On the top you see lights, in this case a high pressure sodium lamp still, relatively older type of lamps. We see in between LEDs. The question is, what would that do? What would be the advantage of this interlighting in between the canopy? Well, the advantage is with in between, there's less reflection to the roof, because if there's light falling on a canopy, there's a lot of reflection, reflected light is lost. You may have a better vertical distribution over the canopy, but there might also be some drop downs. So because maybe the horizontal light distribution is now not so good, maybe the orientation of the leaves starts to change, because that's often what we see. And maybe there gets light on the lower side of the leaf. Is it as efficient? Well, then we can look at the photosynthesis of the upper side of when the light falls on top or on the bottom. That's what we see here. Two curves of photosynthesis versus the PPFD, the light intensity. The black line, that is if the light falls on the axial side, meaning from above, or the red line when the light comes from the bottom. Okay, what we can see, both curves do quite good. So also light from the bottom leads to good photosynthesis, but it's a bit less efficient than compared when it falls on the top. Well, then we did do also with tomato, we did do a lot of experiments where part of the light was on top, part of the light is intercanopy. Usually what we see, if we have intercanopy lighting, we got more growth of the plants. So yes, that could be because of a better light distribution. But what I wanted to highlight also, and in particular because this talk should be about photosynthesis, We see also an acclimation of the leaves. So whether we have interlighting or only top lighting, the light response curves of the upper leaves were more or less similar. So those I do not show here, but this is what we see here in the curve, the light response curves of leaves down in the canopy. And what we see there, that even if we compare them at the same light intensity, leaves which were grown with intercanopy lighting, they have a much higher rate of photosynthesis. So a typical example of if you bring in more light to the bottom of the leaves, of the canopy, those lower leaves will acclimate, and also due to that acclimation have a higher rate of photosynthesis. I hope I'm still running in time. What I want to show here is there's a lot of things here, but it's about intercanopy and top light. I only would like to focus here on light intensity. So if you see in the graph on the lower side, on the x-axis, light intensity, the left side about 250 micromole or 375 micromole. And then we see the yield. Well, with 375, you get much more yield. And in fact, going up with, and these are quite high intensities, what we found was 1% extra light, even give here 1% extra yield. But the point I want to make here, and I do not show you all the result, is that if you would increase 50% more light, and where I said, well, tomato is typically source limited, well, then maybe it would not be source limited anymore. So what we did do here, if we give more light, we also increase the number of stems per square meter. And how does it work with tomato? If you give more stems per square meter, well, you increase the leaf area index, but that's not relevant because that's anyhow at a high level. But with more stems, you get more fruit per square meter, meaning more sinks, and that is needed to make use of all the extra light. So if you do then an experiment on more light, you need to think, as I said at the start of this talk, looking at the source sink balance. And then plants can really use a lot of light, can make use of the extra photosynthesis. Coming back to energy use, first is reduction of energy use. Then we should use the renewable energy and use it when it is cheap. In the Netherlands and many other countries, we may find fluctuations within a day, as shown here on this graph, which can typically is a high price in the morning, high price in the evening, sometimes during the day. There is no cost. Sometimes you can get even money on a Sunday afternoon when it is windy and sunny. There's a lot of electricity supply. Sometimes you can get even money by using the electricity. So prices are not constant, but also the demands of the plant are not constant. The demand of the plant is not the same in the morning and in the evening. So ideally, we have a continuous monitoring. And those things are nowadays possible with, in particular, chlorophyll fluorescence. We can monitor it, and based on the efficiency of the plant, based on the price, on the knowledge on the price of the electricity, we can start optimizing the photosynthesis and with that the growth of the plant. And in these systems, I see that we're going gradually towards an autonomous control, similar as in cars, where we are going towards autonomous driving cars, where we have a lot of sensors, where we have algorithms, where we combine all that information and then can control the climate in the greenhouse. And then I would like to thank you for your attention, and I'm happy to answer questions.
Thank you, Leo. That was super interesting. I'm really intrigued about the autonomous control and optimization of the lighting. I'll give a couple of minutes, couple of seconds for questions to come in the chat. If anyone would like to raise their hand, please do. Okay. So Anton is asking something about the CO2 level. I think it's probably without, I don't know if you want to add to that, Anton, if you'd like to unmute.
Well,
I think perhaps it was I
can probably I can imagine there was a lot of questions about CO2, and I had doubted whether I would also, but yeah, I had only this 20 minutes and I think I used already more time, so I focused a bit on light. It's a very good question. Let's say in Dutch greenhouse, it's already common standard to supply CO2, in particular in wintertime, go to, for instance, 800 ppm. In summertime, let it not fall below the outdoor conditions. But that is where CO2 used to be relatively for free as a waste because we use natural gas for heating the greenhouse. Then CO2 comes with the flue gas. If you're going towards climate neutral, we will not use natural gas. So where does then the CO2 come from? It's getting more expensive, it's getting limited. But CO2 increases the photosynthesis. And this is very important. Usually I say, well, we can optimize everything about the light, but if you have not optimized CO2, well, then you make the wrong steps. It all needs to go hand in hand, but for sure, optimizing CO2, and where, let's say, at least in Dutch type of horticulture, CO2 was, at least in the wintertime, more or less for free. It's going to change. We need to look much more careful at CO2. So I'm not sure what the question exactly is, but at least this is very important. It's getting a very important topic, and also that there are a lot of technologies are, let's say, under development that we can, whether there can be the CO2 can be captured from the air and then supplied in the greenhouse, or maybe that it is used from biodigestation. Yeah, and with CO2, typically what you can see, if you go from 400 to 800 ppm, well, roughly a 30% yield improvement in photosynthesis. So yeah, we should not miss that opportunity.
No, exactly. I mean, I think that's a whole other presentation in itself, right? When you get into this topic. There's a nice question from Davi who is asking, are there low-cost ways to estimate photosynthetic activity, e.g., just measuring light intensity and temperature with more accessible sensors?
Yes, I would say, well, of course, if you measure the light intensity and the temperature, you can estimate what photosynthesis is just based on standard uh, equations. And I would say that can work reasonably well. Um, but of course, that is assuming that is not considering the CO. If you have those two measurements and you don't know what the CO2 is, and let's say you are growing in a greenhouse in summertime and the crop is very well growing and you don't have CO2 supply, well, there's a lot of photosynthesis. The CO2 is taken up, and CO2, where normally 400 ppm outside, it can drop to 200 ppm. Well, then photosynthesis will drop, and that is what you cannot see with a light sensor and a temperature sensor. So maybe then I would say, well, you also need a CO2 sensor for sure. Um, um, and yes, I understand you can measure photosynthesis, but then comes then, of course, at a cost. Sensors are getting cheaper, but on the other hand, nothing is coming for free.
Very good point. Nothing comes for free. There's just another quick question, and then we'll move on to Showcat. In Dutch commercial greenhouses, do they also use far red light?
Limited. So the most common used is only par light. So there are, and then there is, let's say, mostly used is a high fraction of red light, because those LEDs are very efficient in converting electricity into light, and it's good for the plant. You need always a minimum amount of blue, so usually see 5 or 10% blue, and usually, but that has also to do with subsidy. 5% green is typically used in Dutch greenhouses, but yet also use more larger fractions of green and blue. Far red is used limited. Far red, I would say, I very much believe in the use of far red, but then in far red in tomato, there's a strong effect on balancing vegetative versus generative growth. But yeah, I did not include it today because that was not so much about photosynthesis. I see it as a tool for a tomato grower to balance vegetative generative. And you can apply it, but you should not apply it always. It depends on the status of the plants. Something similar with lettuce. We see strong yield increase with lettuce, but you may go over the top and then you give too much, and then you get into other type of problems. But at present, a limited number of growers who use far red.